PharmaPal.
Hypoglycemic effect of Anastatica hierochuntica on male adult albino rats. Welcome to our channel. Today, we're diving into an intriguing study that explores the hypoglycemic effects of 4 medicinal plants on diabetic rats. The research focuses on chromosomal aberrations, blood glucose levels, and the impact on nucleic acids. We'll break down the findings and discuss how these natural remedies could potentially aid in diabetes management. Let's get started. Diabetes mellitus is a serious metabolic disorder characterized by chronic high blood sugar levels. This condition can lead to severe complications affecting various organs. In recent years, there's been a growing interest in using natural products, particularly flavonoids found in plants, to mitigate the side effects of conventional treatments. This study aims to evaluate the effects of 4 specific plants, kafta, dum, samo, and arar, on diabetic rats, focusing on their ability to reduce chromosomal damage and improve overall health. In this study, researchers divided the rats into 6 groups. 1. Normal control, healthy rats. 2. Diabetic control, rats injected with alloxin to induce diabetes. 3. Treatment groups, diabetic rats treated with methanolic extracts of each of the 4 plants. The researchers measured various parameters, including blood glucose levels, total soluble protein in the liver and testes, and the activity of nucleases, which are enzymes that break down nucleic acids. This comprehensive approach helps us understand the plant's potential benefits in combating diabetes. The results were quite revealing. Diabetic rats exhibited a significant increase in chromosomal aberrations, structural changes in chromosomes that can lead to serious health issues. However, treatment with the plant extracts notably reduced these aberrations. Here are some key findings. Blood glucose levels in diabetic rats were drastically elevated, but the extracts helped lower these levels significantly. The DNA and RNA content in liver and testes tissues also improved, suggesting a restoration of normal cellular function. Notably, the activity of nucleases was inhibited in diabetic rats, but treatment with the extracts brought these levels back towards normal. The findings highlight the potential of these medicinal plants as natural hypoglycemic agents. The reduction in chromosomal aberrations and improvement in nucleic acid metabolism indicates that these plants could play a significant role in managing diabetes. The study suggests that incorporating such natural remedies might not only help in controlling blood sugar levels but also in protecting against cellular damage caused by diabetes. Next, we discuss the figures and tables from a study that evaluated chromosomal aberrations in diabetic rats treated with methanolic extracts of 4 plants, kafta, dum, samo, and arar. This study focused on understanding how these extracts can mitigate the negative impacts of diabetes on chromosomal integrity and biochemical parameters. Let's break down the key results illustrated in the figures and tables. Table 1: Chromosomal aberrations in bone marrow cells. First up, we have Table 1, which presents the mean percentages of chromosomal aberrations in rat bone marrow cells after treatment with the 4 plant extracts. Key findings. The diabetic control group exhibited a significant increase in structural aberrations, including gaps, breaks, and deletions. Treatment with kafta, dum, samo, and arar significantly reduced these aberrations compared to the untreated diabetic group. This table highlights the protective effects of these plants against chromosomal damage induced by diabetes. Table 2: Chromosomal aberrations in spermatogonia. Next, let's look at Table 2, which details the chromosomal aberrations in spermatogonial cells. Observations. Diabetic rats showed a higher frequency of both structural and numerical aberrations. The extracts notably inhibited these aberrations, suggesting a potential therapeutic role in preserving genetic integrity. This is crucial as it indicates that these plant extracts may help maintain reproductive health in diabetic conditions. Table 3: Structural and numerical abnormalities in spermatocytes. Moving on to Table 3, we see the results of structural and numerical abnormalities in spermatocytes. Findings. Diabetic rats had a significant increase in total structural aberrations. Again, treatment with the plant extracts led to a marked decrease in these abnormalities. This reinforces the idea that these extracts can counteract the adverse effects of diabetes on cellular structures. Table 4. Blood glucose and total soluble protein levels. Now, let's explore Table 4, which summarizes the influences of the plant extracts on blood glucose levels and total soluble protein in liver and testes tissues. Results. The diabetic control group showed a staggering increase in blood glucose levels. In contrast, the administration of plant extracts significantly reduced blood glucose levels, bringing them closer to normal. This is a promising finding, suggesting that these plants could be beneficial in managing diabetes. Table 5. DNA and RNA contents. Next, we have Table 5, focusing on the effects of the extracts on DNA and RNA contents. Key insights. Diabetic rats exhibited a decrease in both DNA and RNA levels. Treatment with the extracts improved these levels significantly, indicating a restoration of nucleic acid metabolism. This is vital as nucleic acids are essential for cellular function and health. Table 6. Nuclease activity. Let's take a look at Table 6, which shows the activity of DNase and RNase in liver and testes tissues. Findings. The activity of these nucleases was significantly inhibited in diabetic rats. However, treatment with the plant extracts restored their activity, suggesting enhanced nucleic acid stability. This could be a key mechanism through which these extracts exert their protective effects. Table 7. Immunoglobulin levels. Finally, we have Table 7, which examines the changes in serum immunoglobulin levels in diabetic rats. Observations. Diabetes reduced levels of IgG, IgA, and IgM. Remarkably, treatment with the plant extracts significantly increased these immunoglobulin levels, suggesting an enhancement of the immune response. This finding is particularly exciting as it points to a potential role for these plants in supporting immune function in diabetic conditions. In conclusion, the figures and tables from this study provide compelling evidence that the methanolic extracts of kafta, dum, samo, and arar can mitigate the harmful effects of diabetes on chromosomal integrity, biochemical markers, and immune function. Thank you for joining us today. If you found this video informative, please give us a thumbs up and subscribe for more content on health and science. Let us know in the comments what topics you'd like us to cover next.
PharmaPal.